This is the fourth video in a short series on basic concepts in genetics. The second set of terms mentioned is what I'll be talking about in this video. This next terminology set, we're going to start looking at the types of variety. So there is continuous variation, which is what we just looked at in the Camptodactyly, which is a trait that has an expressivity that could be on a sliding scale or a continuum. I think of it as a straight line. This could be from a really mild bend in the pinky to maybe a medium bend in the pinky to maybe a really strong bend in the pinky. It's important to remember that there could be multiple features to the phenotype of a trait. If we look at the Camptodactyly, for example, we could look at a sliding scale that is not just the amount of bend that there is on this person's little finger, but we could look at also, is it one hand that expresses it, or is it two hands? Does one hand express it more than the other two? So there could be multiple sliding scales that are all overlapping for one trait. Discontinuous variation is different in that there is no overlap in the expression of a trait. It's either phenotype A, phenotype B, or phenotype C, or D, E, however many different phenotypes. Maybe there's only 2 phenotypes. Maybe there's 5 phenotypes. The important thing is that there's no overlap in discontinuous variation. Multiple alleles for one gene and a trait being polygenic, I think, are sometimes mixed up a lot. So we're going to compare and contrast what it means to have multiple alleles for a gene and for a trait to be polygenic. Multiple alleles for the same gene refers to the possibilities that exist for that gene. I like to look at blood type because it's a really common trait that most people understand that there are 3 main types: A, B, technically AB, and O. The AB blood type is actually a combination where you have an A and a B allele, and so you're heterozygous. That gets into the ideas of codominance and partial dominance, which we're not really going to cover in this video. But there are 3 possible alleles for your blood type, and that's an A allele, a B allele, or a little I allele, which basically means that you don't have either the A or B antigens on your blood, so you do have anti-A and anti-B antibodies floating around in your blood if you have O-type blood. If a trait is polygenic, that means that there is more than one gene pair affecting the phenotype for that trait. Now, blood is actually a good one to look at for this one as well, because there's not just ABO blood type, there's also a positive or negative. Those are separate gene pairs that affect the different antigens that are on the outside of your blood. If you are Rh positive, it means you do have this antigen present on your blood. If you are negative, it means you do not. Rh positive and negative is a good example of discontinuous variation. You either have it and you are positive, or you do not have that antigen and you are negative for it. So your blood is considered polygenic because it's affected by the gene pair that determines whether you have A, B, AB, or O blood, and it's also affected by the gene pair that determines whether you are positive for the Rh allele or negative. Looking at this question, the question is, a polygenic trait has 3 loci. How many dominant alleles will the offspring of Eduardo and Katya have? First thing we're going to look at is the gametes that Katya and Eduardo produce. All of the gametes that Katya produces will be recessive A, dominant B, recessive C. All of the gametes that Eduardo is going to produce are dominant A, dominant B, dominant C. So we start with our first Punnett square, and you can see the XX parent is on the top, XY parent is on the side. XX parent is contributing 2 recessive A's, XY parent is contributing 2 dominant A's, and then you can see I've already done the Punnett squares for all 3 of these traits. All the offspring will have the genotype heterozygous dominant for A, homozygous dominant for B, and heterozygous dominant for C. So all the offspring will have 4 dominant and 2 recessive alleles. So to wrap up, we're going to look at twins. Monozygotic twins result from one fertilization event, which means one egg and one sperm got together, and then early on in its development, it split so that there were 2 zygotes. Monozygotic twins share 100% of their genes. Dizygotic twins are 2 fertilization events that happen within the same ovulation cycle. This is two separate eggs that are released in the same ovulation cycle, and they are both fertilized by two different sperm. Dizygotic twins share about 50% of their genes. Regardless of whether we're talking about monozygotic or dizygotic twins, if both twins show a trait, that trait is considered to be concordant. If only one twin has a trait, it is considered a discordant trait. The relationship between the concordance of monozygotic and dizygotic twins is studied so that we can try to determine what the heritability of a trait is, whether that trait is more heavily influenced by genetic variants or environmental variants. The next video will consider some questions around the terminology we've just been using.